My name is Bernie Myler. I'm the Associate Dean for Research at Stanford Law School, and I'm just thrilled to be able to welcome you to the second installment of our SLS Faculty Author Series. This time, we're going to be focusing on Hank Greely's CRISPR People. I'm also really delighted that our colleague Michelle Mello, who is a foremost expert on health law in the country, one of the foremost experts, and who's jointly appointed at the medical school and the law school, will be moderating this panel today. We're going to have a number of distinguished speakers whom Michelle will introduce, but I just also wanted to let you know that we will be hosting questions from the audience using the Q&A feature in the bottom of your Zoom screens. So without further ado, let me turn this over to Michelle, and thank you so much for moderating, Michelle.
Thanks so much for having me. It's such a delight to be here to introduce the work of my dear friend and colleague, Hank Greely. We'll talk more about Hank later, but for the moment, let me just set some parameters for the discussion today and just briefly introduce the other panelists who've been kind enough to join us today. We're going to have each of our panelists speak for just about 10 minutes each, the perfect length for the Zoom era. And then we will turn it over to Hank to ask if he wants to say a few words by way of response, maybe 5 or 10 minutes. And then we'll be opening it up for Q&A, and there are some details already in the chat window about how to participate in the Q&A, and I'll go over that again when we open things up. But please have your questions ready for any of the panelists, for Hank, or for all of the above. So let me briefly introduce who's joining us today. First, we'll have Professor Kerry Macintosh, the Inez Mabee Distinguished Professor of Law at Santa Clara University School of Law. She'll be followed by Dr. Kevin Davies, who's a seasoned scientific editor and author of several popular books on genomics and other scientific topics. Next, we'll have Dr. George Church, the Robert Winthrop Professor of Genetics at Harvard Medical School and a Professor of Health Sciences and Technology at Harvard Medical School and MIT. And then Professor Glenn Cohen, who is the James A. Atwood and Leslie Williams Professor of Law at Harvard Law School. So I'm going to begin with Professor Macintosh, again from Santa Clara Law School. Professor Macintosh publishes extensively in the fields of biotech, electronic commerce, commercial law, and contracts. And she's the author of no fewer than 3 books directly relevant to our topic today. Most recently, in 2018, Enhanced Beings, Human Germline Modification in the Law. And prior to that, Illegal Beings, Human Clones and the Law, and Human Cloning, 4 Fallacies and Their Consequences. Professor Macintosh, thank you so much for joining us. I'm going to turn things over to you.
Terrific. Thank you, Michelle. I'm going to change my camera and I'm going to share my screen. Here we go. So we're going to be talking today about CRISPR People. I'm giving an overview with a few comments. And the first thing I want to do is make sure we're clear on the topic of human germline genome editing, or HGGE, is the topic. And Professor Greely defines this as, quote, making intentional changes to the genomes of embryos that will become people whose eggs or sperm will carry those changes. So to continue with the overview, we have part 1, background, which provides the foundation for the rest of the book. It covers He Jiankui, the scientist who created the first genome-edited babies. It talks about the basics of HGGE, the development of CRISPR, and the ethics and law of HGGE before the He experiment. Moving on, we come to part 2, the revelation and its aftermath. And here is where Professor Greely recounts the events leading up to and at the Second International Summit on Human Genome Editing, where Dr. He formally announced the birth of these babies. Professor Greely then goes on to describe how the world reacted, including China, and it's important to note that Dr. He was convicted of unauthorized practice of medicine, sentenced to 3 years in prison, and fined 3 million yuan, which is roughly $430,000. Professor Greely then names scientists and others who knew what Dr. He was doing, but did not report him, some of whom I have on my slide. The 3 academics marked in red actually do work at Stanford. Stanford investigated their contacts with Dr. He and exonerated them from wrongdoing. So that's an important point also. But we will come back to the failure to report in a minute. We then come to part 3, assessing and responding to the He experiment. And this is very important. It's really the heart of the book. Professor Greely does things here. First of all, he assesses the ethics and identifies 5 major problems with the experiment. Quote, a terrible risk-benefit ratio, very questionable consent, inappropriate approval processes, complete opacity, and finally, the violation of what came as close as possible in the world of science to an international consensus against germline genome editing, at least yet, unquote. So that's quite an indictment. Professor Greely then goes on to recommend some solutions: deterrence, disclosure, and humility. Let's unpack that. So there is He Jiankui himself. And for deterrence, Professor Greely urges the scientific community and funders to shun Dr. He, refuse his papers, and reject his projects. In other words, cancel him. We then come to one of my favorite parts of the book, scientific snitching bodies. Professor Greely argues that scientists should have a duty to report illegal, unethical, or dangerous research to these bodies, which could be located in a number of places, as you can see on the slide. Now, I understand why Professor Greely makes this recommendation. He's frustrated with the many scientists who knew what Dr. He was doing, but failed to intervene. But here's my take on it. Nobody likes a snitch, right? And more importantly, nobody wants to be a snitch. Okay, I'm getting some spam here. Nobody wants to be a snitch. If these bodies existed, scientists might decide that it's better not to know too much about what other scientists are doing. Do we really want to create a culture in which scientists are afraid to talk with other scientists about their research? That could reduce collaboration and slow scientific progress. And snitching bodies, unfortunately, will not stop renegade scientists because they will just go underground. We then come to humility. And what Professor Greely is saying here is that science, with a capital S, the scientific community, should show humility by declaring early and often that the public will decide whether HGGE can be used. I have some concerns about this idea also. The message is mixed. That's concern number 1. Because science, capital S, actually does believe that HGGE may have some utility. How do I know that? Because in 2020, the National Academies and Royal Academy created a translational pathway for the correction of mutations that cause serious monogenic diseases. However, on the other side of the debate, religious and political critics want HGGE banned altogether and may persuade the public to agree with them. So if on the one hand, science explains how to use this technology, while at the same time declaring that the public must decide, that's a mixed message. And at best, science looks hypocritical. At worst, science undercuts its own advocacy. Point 2, I'm concerned that this message could discourage scientists who are legitimate scientists from pursuing research that leads to useful therapies. And here I have another slide. I think we all recognize this person, Robert Edwards, who along with Patrick Steptoe developed IVF and received the Nobel Prize in Physiology or Medicine in 2010. Now, when Edwards and Steptoe began research on human embryos, they encountered stiff opposition, but luckily, science, with a capital S, did not condemn them, nor did it loudly proclaim that only the public could decide if IVF was acceptable. So Edwards and Steptoe continued their research, and when they were ready, transferred embryos to women. The birth of Louise Brown is what brought public acceptance. And today, 8 million children have been born through IVF. So we don't want to stop people like Robert Edwards. We do want to stop renegades like Dr. Hay. Unfortunately, I'm afraid they don't care what the public thinks. What might make them hesitate is spelling out all the scientific and ethical challenges, and that is what science has done in that 2020 report. Finally, we come to part 4 of the book, where Professor Greely discusses whether or not this technology is inherently bad. He says no, because the human genome is not sacred and unchanging. Next, he addresses arguments that HGGE is unnatural, unsafe, coercive, inequitable, etc. For the most part, he finds those unpersuasive. He goes on to explain that HGGE, when it comes right down to it, has few realistic uses, and I think he's right about that, that most carriers of genetic diseases will have alternatives, and enhancing offspring is unrealistic for genetic reasons. He also details the process that scientists will have to go through to develop this technology and prove that it is safe and efficacious enough for human use. And it's a daunting process, going to take a long time. Finally, he talks about possible legal responses. He does not believe that an unconditional ban is appropriate. Instead, he discusses regulatory options. So in the end, having said all of this, CRISPR People is more than a tale about technology. It is a warning against scientific hubris. Thank you very much.
Wonderful. Thanks so much for that very helpful framing and summary for those who've not yet had the pleasure of reading Hank's book in full. I hope every one of us will eventually have that pleasure. It's now my role to introduce Dr. Kevin Davies, who is a human geneticist by training, but not by day. He has long been a renowned scientific editor and author specializing in the societal implications of major breakthroughs in genetic technologies. 25 years after he helped launch Nature Genetics, he created the CRISPR Journal, which is the first scientific journal devoted to our topic today, gene editing. Dr. Davies is also the author or co-author of several popular science books, including Cracking the Genome and the $1,000 Genome, as well as DNA, the Story of the Genetic Revolution. And most recently, with the Guggenheim Fellowship, he published Editing Humanity, the CRISPR Revolution, and the New Era of Genome Editing. So he's eminently qualified to offer observations on the implications of this technology. Dr. Davies, I'll turn it over to you. Thank you very much for joining us.
Thank you so much. That was a lovely introduction, and thank you for calling me seasoned as well. I love it. I'm the only non-academic here on the panel, so I'm very honored to be invited to talk about CRISPR People, and so I'm going to try to step back and give you a slightly broader perspective and fill in some of the gaps following Kerry's lovely introduction that you've just heard. Let's see if this will go on, and then we can do that. My own effort to describe and discuss the CRISPR revolution and the whole ethical controversy raised by He Jiankui's actions is called Editing Humanity. It was published about 15 months ago and is available in paperback. And my motivation for writing this book was really the CRISPR revolution over the last few years. I think even people who are law students or law alumni of Stanford have encountered CRISPR and become somewhat fascinated by it. Here's a question that was on Jeopardy that I stumbled upon in 2019, almost predicting the Nobel Prize that came just a year later. when Emmanuel Charpentier in Germany and Jennifer Doudna at Berkeley were awarded the prize in chemistry, really representing a whole field of phenomenal work. Their breakthrough paper came in 2012, published in the journal Science. They weren't able to go to Stockholm because of COVID restrictions, so they received their awards and medals in slightly unorthodox settings. Jennifer received hers in her backyard at Berkeley. CRISPR, when you peel everything back, and that's why we're here, because CRISPR is the technology that enabled this gene editing controversy to take place, began in most unlikely circumstances. It began as a curious observation in microbes, in bacteria and other microbial species, dating back to 1987, when a Japanese team reported a kind of one way to think about this is like a Big Mac sandwich. You can see that these repeated sections in pink, that's like the bread. You can pretty much recognize what the bread is. But the stuff in between in the sandwich, in the filling layer, is perhaps a more dubious origins. You're not quite sure what it is. And for decades, the identity of these intervening sequences was a mystery until this scientist, Francisco Mojica in Spain, ran some computer searches and together with other scientists found that what bacteria were doing was capturing tiny segments of viral DNA and storing it so that they could use it to potentially ward off these viruses in the future. This was sort of an antiviral immune system. This is the textbook diagram that you now see. And so the bits of DNA that are stored in the CRISPR array are weaponized and coupled with these sort of heat-seeking missiles, these enzymes that chew up DNA or cut DNA, hence the Nobel Prize referred in their sort of press releases as Doudna and Charpentier's discovery being the genetic scissors that we're interested in, not just because we're interested in understanding bacterial immune systems, but because we've now, many people, including George Church, our next speaker, have taken these systems and tweaked them and developed them and built on them to now provide a toolbox of gene editing technologies. And here's the last time I sat in George's office interviewing him about some of his work. Indeed, George was one of the first teams to demonstrate that CRISPR could work in human cells as a gene editing technology, which was by no means a foregone conclusion because human DNA is millions of times and much more complicatedly wound and bound and tied up than a simple bacterial genome. But here we are. And here where we are in 2022 is that we are now seeing dozens of trials, clinical trials now underway using CRISPR or versions thereof to treat a growing number of very serious genetic diseases and cancers. And I think the most spectacular results so far dates back now 2 years or more. This is a picture of Victoria Gray, the first American patient to be treated with sickle cell disease in one of these trials led by CRISPR Therapeutics and Vertex Pharmaceuticals. Now more than 2 years since her operation, which used CRISPR to restart or reactivate a dormant gene to help compensate for and substitute for the faulty globin gene that's messing up her red blood cells. She is home, she's healthy, no more transfusions, no more pain crises, really a spectacular result. Has to be replicated many more times, of course, before this is finally approved. But it's pretty miraculous how far CRISPR has come in just the past 10 years. Now, this is what we call somatic gene editing, not to be confused with the hereditary or heritable gene editing, which is the prime subject of Hank's book and makes up about 25% of my own book. This is He Jiankui, who, as you heard in Kerry's talk, traveled to America to do his PhD and spent a year at Stanford in 2011 working in the lab of Steve Quake, renowned biophysicist and on the faculty at Stanford. Steve was one of the first individuals to have his genome completely sequenced and analyzed by a small army of Stanford MDs and ethicists, including Hank you see on this paper, and George Church was on the paper as well. The work was led by Ewan Ashley. So Steve has a reputation for building genetic technologies, and when He Jiankui or JK went back to China after a year to a faculty position in Shenzhen, he took some, with permission, he took some of Steve's technologies to build a sequencing company. But within a few years, his head had been turned and he'd become interested in trying his hand at gene editing. He started coming to America more regularly, returned to America more regularly, giving talks at places like Cold Spring Harbor, posing here with a selfie, interestingly, with Jennifer Doudna. And what inspired him, Hank goes into this in some depth, but one of the trends was that several Chinese groups beginning in 2015 were now reporting using CRISPR to edit genes of human embryos, not necessarily with the intention of implanting these embryos to give rise to genetically edited babies, but nevertheless, this caused quite a storm. New York Times articles, Doudna and others launched international symposia to warn of the potential slippery slope that this technology was going on. But the one person out in front of his skis was JK, who launched an actual trial to eradicate or to inactivate a gene that potentially exposes babies to HIV, and swore Steve Quake, as you heard, and other Stanford faculty and others, a small circle of trust, as it was being called, to complete secrecy, which, for better or worse, they pretty much stuck to. So here's the email that Steve later on shared with the New York Times from JK in October 2018, saying, Steve, great news, the gene-edited baby is born. Please keep it confidential. That news would, if JK had had his way, would have come out with a great paper in a journal like Nature and a famous lecture of his own at his own timing, were it not really for the brilliant sleuthing of Antonio Regalado at MIT Tech Review, who, as I revealed in my book, met JK off the record a month earlier and was able to, couldn't talk about that meeting, but was alarmed enough sufficiently by it that he knew that JK was up to something. And that emerged in his blockbuster story on Thanksgiving weekend 2018, as some of us were traveling to Hong Kong for a bioethics conference, noting that JK was leading a trial where gene-edited babies had been conceived and women were pregnant. They hadn't been born yet, he didn't know that, but the writing was on the wall. So 2 days later in Hong Kong, JK walked into this stage. We thought this was going to be a boring bioethics conference. Most of them are. I think even Hank would agree with that. This was anything but. There were hundreds of press and journalists lined up in one wall of this gorgeous auditorium at Hong Kong University to hear this man defend, because the weight of the world now was on his shoulders as the news of the details had started to leak out, and defend the rationale for editing the embryos of these twin baby girls, Lulu and Nana. He did so rather unsuccessfully. And in a series of news reports that came out in the days afterwards, Kerry mentioned that Hank had sort of 5 main reasons to object to JK's actions. I think you can distill those 5 from the list of 15 that Ed Yong published in The Atlantic, beginning, I think, with number 1. He didn't address an unmet medical need. That was literally the first question that he was asked during his appearance in Hong Kong. And a year later, the paper that he had submitted to Nature and then later to a medical journal that George shared with me, this leaked out, it was published, or extracts were published in MIT Tech Review. A shocking manuscript for many reasons, just one of which I've highlighted at the bottom here. This is the last line of the abstract, where JK says, we anticipate that human embryo genome editing will bring new hope to millions of families seeking babies free from inherited or indeed even non-inherited diseases. So the hubris of the guy really had no bounds. I think for me, one of the most penetrating ethical sort of put downs of JK's work came from in Hong Kong from a senior Chinese bioethicist who said, how could JK and his team change the gene pool of the human species without considering the need to consult other members of the human species? JK left Hong Kong under a cloud. He was whisked back to Shenzhen. He disappeared. He was rumored to be under house arrest. or as I believe the Chinese call it, residential surveillance. And a very enterprising young Chinese reporter from Beijing, working for the New York Times, went to one of the guest residences on campus and spied him lurking on a balcony under pretty heavy guard. And as Kerry mentioned, he was later imprisoned and is now in the 3rd year of his prison sentence. There have been many reports and a lot of hand-wringing about what should the community do, what should we do as a society about in the wake of JK's experiments. I think the most important report thus far came from a commission chaired by Dame Kay Davies at Oxford, no relation, and Rick Lifton, the president of Rockefeller, called Heritable Human Genome Editing, in which they did not close the door completely on applying this technology if it could be rendered safe, and if it was for a very, very serious genetic disease, and if there were no other alternatives such as IVF. But if you do the math, there are very, very few circumstances where that would seem to make any sense. That doesn't mean that everybody is just going to accept those recommendations. There are definitely scientists and geneticists and entrepreneurs out there who would love to get this going, regardless of what the international consensus is. One gentleman who was in that camp is Denis Rebrikov, a Moscow geneticist, Moscow-based geneticist, who in 2019 declared quite publicly that he wanted to follow in JK's footsteps, but this time for a hereditary form of deafness to allow deaf couples to have a hearing-enabled child. The Russian authorities told him to stop his plans, or at least put them on ice. And in the immediate aftermath of JK's appearance in Hong Kong, he received an email that was shared by William Herbert on the Stanford faculty from an IVF fertility clinic in Dubai, expressing interest in learning his gene editing techniques for potential applications. So it would not surprise me if this doesn't flare up again at some point in the future. I want to end on a more optimistic note. The uses of CRISPR should not be confounded by what's happening or what has happened in the hereditary genome editing arena. People like David Liu and George and many others are really doing phenomenal work, expanding, furthering this technology, making it safer, making it more efficient, more precise. And we really look forward to seeing what they can do to eradicate or at least treat patients with a growing number of serious genetic diseases. I interviewed David a year ago for one of our sister journals, Human Gene Therapy. We didn't have a scientific image to put on the cover, but I knew David was a very talented amateur photographer. So I asked him if he had a photograph he might share with us. He sent me a Dropbox of about 100 photos that he'd taken as if for this very purpose. And we ended up selecting the Tree of Destiny, which a photograph that he took in Portland, Oregon. So that maybe is a good note on which to end. I'm very pleased to be on the panel and look forward to the other presentations and the rest of the discussion.
Thank you so much. What a wonderful overview. And I think will be very accessible to many of us in the audience who are not genetic scientists. I want to turn now to Dr. George Church, who I'm honored personally to meet, having shared many years with him at Harvard. Dr. Church, as I mentioned, is professor of genetics at Harvard Med and also a professor at the Joint Program in Health Sciences and Technology at Harvard and MIT. And he leads synthetic biology at the Wyss Institute for Biologically Inspired Engineering at Harvard, where he oversees directed evolution of molecules, polymers, and whole genomes to create new tools with applications in regenerative medicine and bioproduction of chemicals. As those of you who read Professor Greeley's book already read, Dr. Church is among the world's leading geneticists. Professor Greeley calls him a visionary thinker and an immensely creative scientist. In 1984, Dr. Church developed the first direct genomic sequencing method, which resulted in the first genome sequence. And he helped initiate the Human Genome Project in 1984 and the Personal Genome Project in 2005. He's received many awards for his work, including the 2011 Bauer Prize, and he is an elected member of the National Academy of Sciences and Engineering. So pleased to have you with us, Dr. Church. I wonder if you would share some thoughts on the technology here, current efforts, and perhaps some equity issues as well.
Thank you very much. So I think my role is to present on technology and equity and things like that. I don't have any slides. It's sort of in honor of Hank, who rarely, if ever, does that. But the issues, I'd start with the issue of whether there are circumstances under which we would do germline engineering that would impact human health. And I think Hank and many of the people on this panel would argue that there are very few clearly articulated uses for human germline engineering at this point. I think J.K. Hayes' example was among the better ones. It was not perfect in that CCR5, one of the receptors for the HIV, it isn't present in most people, and so it's not something you can solve with in vitro fertilization. It's not something that's easily solved with somatic gene therapy, and indeed the disease of HIV/AIDS is not a solved problem. It's not like there are other solutions, since 2 million people a year die of it. But anyway, putting aside that whether or not that was a good choice, what is the state of play of gene therapy? What is the state of play of germline editing? So I have to admit that my lab probably does more germline editing than most, but it's in animals. So it's in pigs, whose organs are aimed at moving into humans. So it has many of the advantages of germline, and there are advantages that I'll describe in a moment, but none of the disadvantages of affecting the gene pool, as we've heard in the previous talks. It has the advantages of germline. Let's start with that. Is that they are the delivery is to every cell in the body very efficiently and without too many errors. I mean, it basically is as good as you can get for delivery of every cell in the body. It also has the advantage that you can do quality control on it. You can do it very early in embryogenesis. Many diseases, and in fact, everything about us is determined early on. And you can do multiplex editing. Multiplex editing means that you can change many genes at once. And again, that's because of the quality control that you can do on the cells that are destined to be the germline of, let's say, the pig. Now, once you have, so some of these pigs have been engineered at up to 42 different places in their genome. And this also bears upon the question that's come up a couple of times already today about the safety. But you can safely engineer 42 different changes in the germline of pigs, and they're quite healthy. We've done this in many strains of pigs, and and the organs from these pigs are already in preclinical trials in 3 hospitals. Plus, one case has already been made a heart transplant into a human, which has been in the news considerably recently. So these are things where they've been vetted, so to speak, in pigs, that they're shown to be healthy. The pigs can go through multi-generations, and they can donate these organs to humans. These pig organs are enhanced in a certain sense. They are immunologically privileged relative to both pig and human organs, and that they are sort of universal donors. They are also enhanced in the sense that they are resistant to a wide variety of human-specific diseases, including HIV. And they could be even enhanced even further. They could be senescence resistant. They could be resistant to a wider range of pathogens, and so on. So the idea of avoiding enhancement, I think, is a little misguided. Almost all of our technologies are enhancing in some respect or another. What I think we have a very special relationship to one particular form of inheritance. There are many forms of inheritance. We inherit, education, religion, technology, money, all sorts of things, but we don't put that in the same category. And we might inherit generation after generation, and indeed more faithfully than we inherit our genes. If you look, kids, often siblings and parents don't always look alike or act alike or share much of anything, but they will faithfully inherit some things for centuries that are non-DNA. Nevertheless, we accept the societal consensus on the specialness of the germline, and we try to do the best we can by doing somatic engineering, either via the germline of the pig or via DNA or editing enzymes or various mechanisms throughout the life of the human. And this can go earlier and earlier. So when we can get to, in fact, many serious genetic diseases have already done so much damage by the time the child was born that there's really no practical surgical nor somatic gene therapy that will work at that point at time of birth. So you need to go earlier, and you could do genetic gene therapies, including editing, prenatally. You could do it in the fetus or the embryo, as long as you don't touch the germline. So I think we are not generally concerned about the safety of embryos and fetuses so much as the safety of the germline. It's treated very specially. It's sometimes called exceptionalism. That is, and to some extent, when we talk about safety, in this particular case, we now have 2 or 3 children that have been born with germline CRISPR editing of the CCR5 gene. And they're probably healthy. I mean, we haven't really heard reports, and I think that's a bit of, it's a continuation of the non-transparency in this case, which I think a number of us take offense with that lack of transparency in general, certainly prior to the announcement. But if you compare it to other new categories of therapies, for example, monoclonal antibodies, in their first trials with monoclonal antibodies, there were 4 patients, and all 4 of them had very serious consequences, almost death. The first trials on gene therapy, somatic gene therapy, which we now consider okay. In fact, there are many FDA-approved ones, but back in the year 2000, there were some deaths for 2 different reasons. One of them was a cancer due to the LMO2 oncogene, and the other was immune reaction. So what's interesting here is this case seems in certain ways, in terms of the health of the children, to be more in the category of the in vitro fertilization that we heard about earlier with Louise Brown than they do in the category of monoclonal antibodies and gene therapies, which are marvelous medical revolutions, but did involve some very serious consequences for the initial patients. So I think we need to put this in perspective. I'm not supporting anything that JKA did, but I think we need to just kind of get a perspective on where we stand in the progress of this field. So I'll just say that technologically we're moving forward quickly, and I think one of the top ethical issues that really hasn't come up much. I mean, we've been talking about safety and efficacy, which is really the FDA purview, but the FDA doesn't always weigh in on the long-term consequences, the overall wellness of the individual. They'll approve things like statins that lower your cholesterol, but don't necessarily improve your quality of life or your length of your life or so forth. And then third and most important is equitable distribution. Now, one of my objections to gene therapy, even though I've been involved extensively, my lab has produced a number of not just CRISPR, but delivery vehicles and non-editing gene therapies. One of my concerns from the beginning, and one of the things that we work on is reduction of technology costs to make it more equitably distributed. And in the case of gene therapy, this has undergone a revolution. Just like the cost of gene sequencing, reading the genome has gone down from $3 billion to $300 and improved tremendously in quality to something that's clinical. The same thing has happened with gene therapy in a very special case, which is most rare gene therapy. Gene therapies for rare diseases are on the order of millions of dollars, $1 or $2 million per dose. The recent one that many of us have taken, in fact, the 4 top vaccines for COVID-19 are all gene therapies. 2 of them are adenovirus, so the Sputnik and the AstraZeneca are adenoviral delivering double-stranded DNA, and then 2 of them are lipid nanoparticles delivering messenger RNA. The point is that these are capable of getting down to $2 a dose without losing money. $2 a dose is getting to be down from $2 million for some of the rare ones. What they have in common, I think, is the very large market size, and they also got approved very quickly. So these are new categories of drugs, and they got approved in roughly a year. So I think some of these things are indicating the direction that this technology is going. We won't be seeing any germline directly into human germline until there is a very compelling case made, and that case will include extensive use of animal germline delivered into humans, maybe combine the blood cells and various organs, plus greater confidence in multi-generation inheritance in germline engineered animals, and then success of lots of somatic gene therapies. When we have those 3 things, we might assert 4 things, including the argument for a medically compelling case, then we might consider human germline. Thank you.
Thank you so much. It was a really fascinating tour of where things are heading and starting to get into some of the cost issues that I think we'll probably want to return to in Q&A. Before we do that, we have one more really wonderful speaker to hear from, and that is my friend and colleague, Glenn Cohen. Glenn, as I mentioned, is James Atwood and Leslie Williams Professor of Law at Harvard Law School. He's also the faculty director of that law school's Petrie Flom Center for Health Law Policy, Biotechnology, and Bioethics. Professor Cohen very quickly became one of the world's leading voices across the fields of bioethics and health law. He is just astonishingly prolific and has an incredibly agile mind across a huge range of topics. His over 200 articles and I think now more than 18 books span topics as diverse as medical tourism, assistive reproductive technologies, research ethics, organ transplants, and more these days, issues relating to use of big data, AI, and health information technologies. I've had the pleasure of collaborating with Professor Cohen on several occasions and really appreciate how he can offer incredible insights on really any topic in the realm of health law and bioethics. And he's also part of the production of bioethics scholarship in his capacity as one of the journal editors of the Journal of Law and Biosciences. And serving on the steering committee for ethics of the Canadian Institutes for Health Research, which is the Canadian NIH essentially. So I'm very pleased to welcome Professor Cohen into the discussion and can't wait to hear what you have to say.
Thanks so much, Professor Mello. I also don't have any slides. Thank you also to Professor Greeley for the opportunity to fetch you for this book. This is an unusual but wonderful opportunity. It's unusual because I'm almost always invited into panels where I mostly disagree with people, whereas there's almost nobody in the world whose take on these issues I agree with more than Hank's. We do have a few subtle areas of disagreement that I'll speak about shortly, but I can't spend the whole 10 minutes just praising him, but I want to at least start by itemizing all the ways that in particular part 4 of this book is really, really good and really, really right on. And I'll say, for those of you in the audience who haven't read this book or worried that it's going to be boring, it is incredibly readable, which is not something easy to do while being accurate about the science. Okay, so what is it that Hank gets right that I want to particularly praise him for? The first most important wisdom, I think, in Hank's approach is to reject what is sometimes offered as a way of framing this question that he rightly views as silly, a view of germline gene editing as all bad or all good. Indeed, he begins chapter 12, titled, "Could Human Germline Genome Editing Sometimes Be Bad?" With the words, "Of course, what technology can't be?" Part of this is Hank being Hank in the writing, but the deeper point, I think, is to de-exceptionalize HGGE. At one point in the book, he mentions smartphones, and I often tell people that when you ask me which technology is at high risk of radically reshaping a society with large follow-on ethical complications, a genie out of the bottle, it's actually the iPhones, which had zero pre-clearance, not the gene editing, I think, where we've had wonderful NASEM, Royal Academy reports, discussion of international treaties. So one thing I like about the book is it gives gene editing its right due and places it in the right place in the pantheon. I think this portion of the book does a great job pouring some cold water on some of the techno-optimists, making the point that even a decade from now, when the techniques of HGGE might be better, it's unlikely to be a go-to for most of our needs. A combination of PGD and somatic editing will be preferable for a large number of possible uses. But there will still, as he points out, be some uses left. I think Hank finished this book before the September 20 NAS UK Royal Society Heritable Human Genome Editing report that Professor Macintosh also mentioned. And I think their attempt to divide uses of genome editing into 6 categories was pretty good. Here, I'll just quote from a paper I did with Dr. Adashi in the Hastings Center report describing these categories. The report describes 6 categories of potential uses and endorses the possibility of, quote, a responsible translational pathway for initial uses, unquote, for what it calls category A uses, which are, quote, cases in which all of the prospective parents' children would inherit the disease-causing genotype for a serious monogenic disease, that is a monogenic disease that causes severe morbidity or premature death, and also for what it calls category B cases, that is, quote, cases in which some but not all of the prospective parents would inherit would inherit the pathogenic genotype for a serious monogenic disease, unquote, which it describes as a very small set of circumstances. For these category B cases, it says we need reliable methods to be developed to ensure that no individuals resulted from embryos that have been subjected to potential adverse consequences of genome editing without the potential benefit. Such methods would depend either on identifying zygotes or embryos with the disease-causing genotype before performing the edits, or on excluding from transfer embryos that had needlessly undergone editing in category B. Now, the report therefore rejects the claim that genome editing is never necessary because adoption or gamete donation or PGD or somatic alteration is available. And I think maybe Hank undersells that his position and mine and that of this report is at variance with the claim of many who seem to suggest we don't ever need genome editing. In particular, I think one key question that is a little under the radar in the book is the strength of a rights claim towards genetic parentage. Now, all these outcomes are achievable using third-party gametes. That is to say, we could insist that instead of genome editing, these people use gamete donation, sperm donation, or egg donation. To say one has a prima facie right to be a genetic parent to one's child does not necessarily mean that this rights claim is indefeasible. That is, a society could go technology by technology and say for this one, The reasons against uses are sufficient to defeat that claim. That's not where Hank is, that's not where I am, but I want to surface this as I think an interesting and live issue. I also want to praise chapter 14 called How to Test Human Genome Editing, which gets deep into the weeds, just how complex it will be to run preclinical and especially clinical trials for genome editing in the FDA pathways, and pushes on the question of off-label use post-approval, which is, I think, something that not enough people have thought about. I think he and I are very much in agreement that the FDA pathway, especially in its post-approval space, is actually an awkward fit for how we would like to see genome editing regulated, and that the HEPA in the UK's ability to license particular facilities for particular technologies would be something desirable, but potentially not politically feasible in the United States. All right, so that's the praise section. I do have some minor points of disagreement or more questions. First, surprisingly, one is on religion. Hank doesn't spend a ton of time on it, but he does sound some skeptical notes about the religious traditions as to their opposition to genome editing, saying as Judeo-Christians, quote, one is left to try to distinguish without clear textual support between the permissible deviations from the nature, ... and impermissible ones. It's hard to find any clear distinctions between human germline genome editing and other modern innovations, unquote. And then he says later, quote, don't get me wrong, if, let's say, an Amish person believes that God is fine with him using power tools, but not with him driving, I have no objection to his living those beliefs. I do object to his imposing those views on others who do not hold them, unquote. Hank and I think, are both, broadly speaking, libertarian leaning about things, including genetic technologies. But I do think it's worth pushing on the role of religious views in making public policy in this space. I think I'm more of a classical liberal than he is, at least in this book, in being willing to entertain more comprehensive views of the good life, including religious ones, in the dialogue. This brings us back to the ideal of public reason and what a viewpoint needs to consist of to get at a seat of the table in a deliberative democracy perspective or experience. So for me, Kantians and Christians might more comfortably sit side by side in these discussions than perhaps for Hank. In other areas like debates on the wrongfulness of embryo destruction or views of when personhood begins, I'm not sure it's as easy to separate out the secular and religious moral objections, nor whether we should do so. To put it starkly, imagine we were to have a deliberative democracy exercise for our country to set human genome editing policy. I'm not sure I would tell people at the start to leave their religious views of human genome editing at home. And I'm not sure what Hank thinks. There's times when I think he reads as though the answer is yes. Other times I'm not so sure. Second, I want to highlight some disagreement on the enhancement treatment distinction. So Hank has written a bunch about that elsewhere, and I'm not sure how much of this is just him pulling his punches a bit in the book. Many, but not all of the major reports treat this as a useful line and want to draw distinctions on either side of it. I'm in the camp that's much more skeptical about the treatment enhancement line, which is often drawn around the concept associated with Norm Daniels of species typical normal functioning. Treatment being what returns us to what is species typical normal functioning, enhancement what takes us beyond it. As a moral and conceptual matter, I think this framing suffers from a serious baseline problem. And I think Hank kind of indirectly pokes the bear a bit on this in his sections attacking the idea of a common genome and the importance of that idea, pointing among other things to vaccination. My own view is that these large categories of enhancement and treatment are more obfuscating than helpful. And instead we ought to look at a series of more specific lines that might or might not matter for particular technologies. Safety, whether we're talking about raising people to levels that are currently attainable versus introducing new things into the genome. Reversibility versus irreversibility. Equality promotion versus inequality exacerbation. And I think Hank is broadly sympathetic. What makes it a bit surprising that he seems more congenial to the enhancement treatment line drawing in the book than I would have suspected. Okay, finally, one more. I think one area where Hank says less than I would have expected is as to future generations and the effects thereon, and what consent conditions, hypothetical consent conditions, contractualism or whatever, should look like. Some of his discussion of when somatic versus germline alterations are appropriate and the costs of redoing it generation by generation if we did it somatically nudges in this direction. But my own view is that on some views of how radical a germline alteration would be as to disrupting genetic identity, the whole concept of person affecting harm and benefit, that is, that people in the future would be harmed or would be benefited from a technology like germline gene editing, might be problematic because we're not talking about making particular people better, but replacing them with other people. And the reasons for replacement might be quite different from the reasons for making somebody harmed or benefit. Okay, lots more to talk about, but I'll stop there. But thank you, Hank, for writing this great book.
Thank you so much for those reflections. And with those provocations, I think it's an ideal time to turn things over to our honored guest, Professor Greely. Let me just say a few words about him for those who haven't had the true pleasure of having him as a professor at Stanford. In addition to being the Dean F. and Kate Edelman Professor of Law at Stanford, he is Director for Center of Law and Biosciences here at the law school. And in the course of his over 40-year career, I think has intervened intellectually in just about every scholarly and policy debate about important developments in the biosciences that has erupted. He is especially prolific in his writing and advising in areas of genetics, neuroscience, assisted reproductive technologies, and stem cell research. But again, you will find Professor Greely's intellectual footprint over most of the major science committees and thinking about bioethics over the last almost half century. Professor Greely is an elected fellow of the American Association for the Advancement of Science. And in addition to this wonderful book on CRISPR People, he's the author of the provocatively named The End of Sex and the Future of Human Reproduction, which I also recommend to everyone. As you know, if you've read the book, he is just incredibly adept at analyzing and communicating about the complex legal, societal, and ethical aspects of developments in biotechnology. And I personally really admire his ability to constantly stay right at the cutting edge in terms of the topics that he's working on and his understanding of the science, and also just to shoot from the hip, which makes him an ideal person to communicate with this broad audience about this complicated topic. So, Professor Greely, I'm going to turn it over to you to say whatever you might wish to say for the next 5-10 minutes.
Well, thanks, Michelle. And first, thanks to Bernie and Stanford for putting this on. Writing a book, it's like launching spacecraft out where you only hear occasional reports for the first few months and then nothing but deep silence. So to actually get the sense that 11 months after publication, something's still going on with this book is quite satisfying. Michelle, I really appreciate the introduction and you pointing out that I have sort of intellectual ADHD or dilettantism in jumping from thing to thing, but hey, it's something that I really enjoy, being able to cover lots of topics. I really enjoyed and really appreciate all of the commenters on the paper. I think what I'd really like to do is take them all out to dinner and over several bottles of wine, discuss this at much greater length, whether the wine is enhancing or dehancing, Glenn and I can argue about. But again, it's nice to see that this product still is alive and doing something. Let me say a little bit about each of the speakers' comments. Kerry, yes, I think the snitch issues and the humility issues both have their problems. I'm not blind to some of the concerns that you raised, which I think are reasonable. On balance, I think increased snitching, though a difficult thing to do, would be a good thing. And I think increased humility is almost always a good thing. But humility by science, I think we are seeing in part across the world a backlash against expertise that I find deeply distressing and that is killing hundreds of thousands, if not millions of people. But part of that backlash, I think, is that some folks have the sense that science with a capital S, thinks it's always right and is eager to boss people around. I think it's useful for science to be clear that it is ultimately part of a society, part of a culture, and has to abide by its rules. Kevin, I really enjoyed your book. One of the interesting things about this is a whole bunch of books came out at about the same time. Kevin's book, my book, Eben Kirksey, a STS scholar, Science Technology Studies scholar, has a book. Françoise Baylis had a book out. Kerry's book came out. And then the however many pound gorilla Walter Isaacson's book came out at the same time. And I think Kevin probably devastated all of our royalties. If each of us could only sell 1% of what Isaacson did, we'd be much richer. But Kevin's, I thought, was a unique contribution. And again, most of Kevin's really wasn't about the He Zhonghui episode fiasco, I think, which mine focused on, but it's a really great introduction to the history and the science and the many potential uses of CRISPR. And I want to make it clear, I am a huge fan of CRISPR, but all technologies can be used well or can be used poorly. To George, I do think in retrospect, I wish I had been a little more open in the book to other potential uses. And this goes in part to something Glenn said as well. I do think there aren't going to be any really powerful, there'll be very few, but some powerful, compelling uses for this, mainly with people who want to have kids, whose kids would necessarily have serious, terrible genetic diseases unless gene editing was involved, whose genetic children would have them unless gene editing was involved. But I think I do believe, and this doesn't come through enough in the book, that given time and a track record of success and safety shown not just in clinical trials and preclinical work, but in a decade or more of use in difficult cases, in compelling cases, we could think about using it as sort of, preventive deletions of autosomal recessive genes so that alleles so that people wouldn't be carriers. I don't think that's compelling right now, but it's something that I think we could do if it felt safe enough and if access, the cost was low enough and access was broad enough. I think preventive deletions to avoid other diseases like CCR5 and HIV could be a good thing. Both of those, I think, though, would require more understanding of what the consequences are of taking that out. There are some indications, pretty preliminary, that losing CCR5 may make you more susceptible to bad effects from influenza or West Nile disease. I don't think they're definitive. I think they're provocative. But the fact that pretty much all vertebrates have CCR5 genes is some implication that it's probably doing something useful. So we might want to do that in the future. And then talking both to George, but also to Glenn, the enhancement issue. I'll come back to the enhancement issue, but I'm not closed to enhancements totally. I do think the safety questions for a long time are going to make any kind of non-compelling uses less interesting. The exceptionalism point, I am glad to try to put a stake in the heart of any kind of human germline genome exceptionalism. But I do think, and this goes more to George's comments, I think I am a believer in making babies exceptionalism. And I do think we need to be especially careful about potential negative consequences when we're making babies. It is deeply unfortunate when somebody in a human gene therapy trial ends up dying as a result of the trial. But if that person was consenting and other options, was otherwise looking at a very serious disease, that makes it a little less of a tragedy than if a baby who had no choice in the matter turns out to be disabled because of some way we went about trying to create them. So I do think we should be exceptionally careful when it comes to methods of making babies, even though as a parent myself, I can't say that I'm sure that my wife and I were thinking through all the risks very carefully before we did it. And then Glenn, we definitely need to get together and talk at length, but I appreciate the things we agree on. The ones we disagree on, I'm not sure how much we disagree. I didn't get into the rights claim to genetic parentage, in part because you'd written about it so thoroughly, and the late lamented John Robertson had written about it so thoroughly. I agree with it, but that wasn't what this book's primarily about. And I think it's evidence of a bit of a tension I had in writing this book. Part of me felt like a lawyer who's writing a memo for a client. Here are the pluses, here are the minuses, here are the issues, you make the decision. It's your decision ultimately. But part of me also has substantive views on some of these things. And I'm not sure I managed that distinction or that perspective quite as thoroughly as might have been good. I agree with you on the enhancement treatment distinction. I think the book makes me sound less open to enhancement than I actually am. Right now, I think most of the things people think of as enhancement and super babies, we just don't have a clue about how to do. But in the long run, I think the enhancement treatment distinction, though in a very fuzzy sense, useful, isn't really very rigorously useful. And I think if you want to define humans as one characteristic of humans, it's that we've always been trying to enhance our lives and enhance ourselves and our environment and our children. So I'm with you on the enhancement issue, and I don't think it came through quite as much in the book as it should have been. And I think in the interest of letting other people talk, since I already had 300 some odd pages in the book to talk, I will shut up there and turn things back over to my friend and colleague, our moderator, Michelle Mello.
Thank you so much. Let me invite all the panelists to rejoin us on camera at this stage. I'm going to open the floor for questions. You can submit your questions using the Q&A function at the bottom of the Zoom screen. And I will just say that please be unafraid of asking questions of clarification about the science. We have a wide range of folks on the call today, and I know that everybody wants to make sure that there's an understanding of the technologies that we're talking about here, as well as their implications. So I'm happy to take any and all questions, and also to have our panelists ask questions of one another. But I'll get the ball rolling. I'm in public health, and so I tend to think about the impact of innovations on the population level. This is a technology that all of you have talked about as being being steered towards individual families in very severe cases. And maybe I'll ask Dr. Church to be the opening commenter on this, but I'm interested in everyone's views. Can you ever envision a time where the cost of this technology would be low enough and the discoveries broad enough that we could think about intervening at a population level? For example, what if we found a tweak in the genome that would make humans impervious to all coronaviruses? Is that a future that you can envision from a technical standpoint, Dr. Church? And how would you think about whether it's a future we would normatively want to be moving towards?
Yeah, thank you. I think that's an excellent example, and it applies both to coronaviruses and to HIV. Is that Hank pointed out that these proteins that they bind to, the receptors, are conserved in all other mammals. So how could we get rid of it? Well, the only reason we talk about getting rid of it is because of the laziness of the CRISPR community, which is CRISPR is mainly used for genome vandalism. It's not used for precise editing. But if we replace the human CCR5 with the CCR5 from some other organism, or replace the ACE protein that is the receptor for coronavirus with one that's from an animal that doesn't get the serious disease that we get, we might fairly simply make them resistant to HIV and CoV-2 without making them susceptible to West Nile or flu, because we haven't deleted the gene, we've just altered it in such a way that it is no longer susceptible to these human-specific viruses. So the question of cost, part of the reason I celebrated the low cost of the 4 vaccines that are the top vaccines, and also all are gene therapy based, is that yes, I think the price can come down to $2 per dose. And since gene therapies tend to be a once in a lifetime thing, and germline would certainly be once in a lifetime, yes, I think they could be equally distributed, they could be safe, and they could be extremely effective against many viruses or many infectious diseases more generally.
Well, so let me open the floor. Thank you so much for that technical grounding. I'm actually delighted to hear that my question wasn't out in left field scientifically, but let me open the floor for commentary on the normative aspect of the question. Is like population interventions with germline gene editing something you would support moving towards, and under what conditions? Maybe can we start with Dr. Davies?
Well, I remember when I was first, I think, starting to research my book, and obviously if you start writing a book about genome editing, you immediately gravitate to George's writings and talks, and he for many years has shown, even really before CRISPR came into its own, would present a slide of potential genes or pathways that we might want to interfere with for enhancement purposes. And I remember the norovirus receptor was one that he had up there very early on. So you cross that out and you put coronavirus, whatever the final receptor is shown to be. That is it's a very intriguing place. But if we're talking about germline population editing, from just a practical sense, at least based on the way that we do it now, there may be more convenient ways down the road, but that it seems it's not so much price, but just I don't see the whole population running to their local IVF clinic in order to try to bring this about. I'm sure George has more less intensive ways to potentially do this. The other thing about pricing, just very briefly, is we, as I mentioned, we're seeing great progress on sickle cell and other genetic diseases, and some of these will, I think, be approved in the coming years. But the pricing question remains a major problem, not just for CRISPR gene editing, but gene therapy. And indeed, we know we're all getting, in some ways, ripped off by the exorbitant prices of drugs and therapies in this country, and CRISPR, for all of its wonderful technology, doesn't automatically change that equation. So the companies leading this are still beholden to their shareholders, perhaps more than their patients.
I'd love to come back to that issue in just a moment. Before we do, are there any other comments on this broader question about population level editing?
I do think that Kevin pointed out one thing that is likely to be a constraint, at least for a while, and that is IVF. And I think the most plausible ways of doing germline editing will involve IVF, either in terms of making the embryo or checking the embryo to make sure that the edits were properly done. And IVF is a big rate-limiting step. It's unpleasant, it's somewhat risky for the woman, and it's expensive. My earlier book, The End of Sex and the Future of Human Reproduction, talks about making germ cells, eggs and sperm, from stem cells, ultimately from skin cells. Glenn has also written about this. Its crypto-Orwellian acronym is IVG, in vitro gametogenesis. And I think that's likely to happen. That would bring down the cost and difficulty of IVF in a way that might make broader scale use, population-wide use, or population scale use of human germline genome editing more plausible. But that's at least a decade off, I think.
So let's turn to this question of cost. One of our audience members wonders, when we think about these types of enhancements, isn't it inevitable that the distribution is going to be inequitable? Cost obviously is coming down, but what do you see as the barriers to ensuring equitable access to these kinds of technologies across groups of patients?
Maybe I'll start if that's okay.
Yeah,
I'd like to say also that it's a very lumpy terrain in the following respect. So there's ways in which you might view something like the COVID-19 vaccine as an enhancement rather than a treatment. One could imagine the equivalent against cancer. Imagine we were able to produce generations that never got cancer. There are many sorts of enhancements where their value is not positional, but absolute, in the sense that we all benefit the more people who get them, and therefore we have every incentive to subsidize them, as opposed to positional benefits, right? And part of what I want to say about this is that just anything that's universal, the more, so if you're thinking about what to create as a scientist, right? So you're either an altruistic scientist or you're a venture capitalist. you've got a market that's the entire world, that the entire world can benefit from something and have purchasers. There's a lot of attractiveness for that rather than something that's incredibly bespoke and being catered to the very rich. When I worry about what makes our societies less equal, I worry about all sorts of things that have nothing to do with genetics. I worry about SAT tutoring more than I do about gene editing. And so for me, but if you are concerned about this, I think the solution is let's focus the research dollars, let's focus the support for departments on the kinds of changes to the genome that benefit all of us, not those that make the genetically rich even genetically richer.
Professor Macintosh, is there anything about the development model for these technologies that presently contributes to their cost?
Well, I certainly think that IVF itself, which would be the foundation for these technologies, right? You need to be able to edit the embryos. IVF itself is very, very expensive. A lot of people can't afford it already. So, you know, how they're also going to be able to afford these add-ons where the embryos are edited, I'm really not sure unless somehow there's going to be insurance coverage. And currently there often is no insurance coverage for IVF. You see people who are, you know, unable to buy a house because they put all their money into IVF treatments, which may or may not have worked, or they had to sell their cars in order to afford IVF, or they've got a GoFundMe going. So in terms of universal access to this technology, I'm not at all optimistic.
Any other comments from other panelists on this? Yes, Dr. Church.
I just want to quickly note that we need to disentangle the current technology from future technology, because these things change very, very quickly. And so I am not convinced that IVF is the only way to go on this. We know how we're getting better and better at delivering cell therapies. And if you think of gametes as cell therapies, they could be delivered in a variety, in a very simple way that does not involve. So you're delivering gametes to the mother and the father rather than delivering eggs. Embryos. And so you don't really need the IVF clinic if you can deliver either deliver the genes to the gametes or deliver the gametes themselves, the gamete precursors. So I think we need to be open-minded if we're going to have, you know, especially in these forward-looking conversations, we need to consider something that's really not that far off. Now, when Hank says something is 10 years off, that's a very safe prediction, because the average time for getting FDA approval is 10 years. The exception being COVID-19 vaccines, which only took 1 year, and maybe everything will get shorter in that if it's sufficiently significant. Anyway, that's my view on the IVF question.
I'd like to follow on Kerry's comment for a second and just name drop a great book called The New Eugenics by Judy Darr, who's the dean of Northern Kentucky Law School, who points out that because IVF is expensive, we're effectively preventing poor people from reproducing if they've got fertility problems that can only be overcome through IVF. Because if you're poor in the US and you're infertile, good luck, you're not going to be able to get IVF. So I think that's a huge part of of course, the shameful lack of any kind of universal, decent health coverage in the US.
Let's turn to the topic of regulation. We have a few questions circling around this theme. Hank, in your book, you suggest the need for more snitching, or maybe we would more politely call it whistleblowing. And certainly there are other industries where we can point to whistleblowing as having been a very effective mechanism for surfacing at least some of the worst abuses in the system. The one that's maybe most salient for us right now is Facebook and big tech, but there are many others in healthcare and outside it. On the other hand, with a lot of our professions, we have a lot of evidence that individual members of the profession are very reluctant to turn in colleagues, even when, for example, in medicine, evidence of impairment is abundant and harm has already occurred. So how should we think about the role of self-policing in science here versus a more invigorated formal oversight system for human subjects research, and possibly even reaching into the kind of preclinical work that Dr. Church's lab does in a way that transcends our current animal research regulations? Professor Cohen, you want to start?
I think Hank's book chapter is really one of the very best. That, and I would say the 2020 NAM/NAS Royal Academy report in this respect. There is this difficult, so earlier there was a comparison drawn to IVF, which I think is a very interesting comparison that's wielded by different people very differently. There is a moment when you move from preclinical to clinical, or to first in humans in clinical trials, where there's just a lot of risk, and you can do everything you want on the front end to try to reduce that risk, but we really don't know, and there's no way to kind of future-proof it. And I think part of what's interesting, if you've read about the history of IVF, is that IVF grew up in a period of a different regulatory structure, and also one, and Professor Mello, I think you will particularly appreciate this, where the line between the practice of medicine, which FDA does not normally touch, and drugs and biologics was more forgiving, I think, in a way than it is for these technologies. So I do think that there's just this very difficult question of when are you ready? And we're all very glad that IVF, they took the step. It's all ended fairly well, although actually the long-term, like 100-year consequences of IVF, there are still some open questions. But I think Hank is like, sets up exactly what you would need to do step by step. But there's a moment where you just have to hold your breath. And I think that's an uncomfortable truth, in my opinion, to kind of, face.
Other thoughts on the respective roles of self-policing versus formal regulation here?
I think I would just say, I'm sure Hank has had many, some on, but many off the record conversations with some of his colleagues who were in He Jiankhui's circle of trust, which actually proved to be quite a big circle at the end of the day. Many big names in China knew what was going on. Several prominent American scientists knew what he was planning and then what he was doing. So he wasn't quite the rogue operator that he was widely perceived to be. And I think most, if not all of those people, certainly here in the US, would probably, if they had to do it again, would say, we discouraged him quite strongly, but we respected his request to stay in confidence. I think they wish they could have that. If they could do that over again, they might have gone to the New York Times or the director of the NIH. I don't know where you go. I think we're still trying to figure out that. So I think if there's some good to come out of the JK affair, it's that I think it's woken us up to the need to be a little more vigilant in the public interest.
Dr. Church?
Yeah, I just wanted to weigh in that I'm happy to be a snitch, but I don't think of it as being such. It is really just raising consciousness about what could be happening. It's not saying so and so should be thrown in jail. It is saying there's a group of people that may not be aware of the consequences of their actions. So I actually have been a snitch in that regard numerous times, and my favorite go-to is the FBI. Quite frankly, they're increasingly sophisticated in biotechnology, and they have invited the entire synthetic biology community to just report anything that they think is of concern. And no one has been thrown in jail because of my snitching, but but I'm sure there has been some investigation, and I don't think this is interfering with their inherent human rights. Nobody forced them to become a synthetic biologist, and to Kerry's point about having a snitch culture drives everything underground. I'm specifically talking about the underground. Is if there's something that's not in the public domain that you know about, that's when you should be going to the FBI or some other agency. I think that really everybody had an obligation. I would hope that if J.K. He had said this in my presence, I would have done what I did in a number of other cases, and I would have gone to the FBI.
Thank you. Well, one of our guests is wondering about where the public lies in all of this discussion of who gets to decide what's permissible, and asks, when people say the public should decide bioethical issues like this, who are they talking about? So are we talking about voters? Are we talking about their elected representatives? And how would you suggest that we think about involving the public, if at all, on this particular topic? Throw that up to anyone who might like to take a stab.
Just a quick one. I think a lot of us do try to engage the public, and the problem is not the reluctance of the scientists to have public engagement. I don't think we need to self-police, other than our access to the underground, non-transparent components, where we really are the ones that have the access to it and are the best position to snitch. But engaging the public is not easy. They're busy with lots of other things. Some of the best efforts I've seen for engaging the public is pged.org, where they go to congressional aides and to Hollywood and to other to high schools, where there is where you can reach millions of people. That's what it takes, and I don't think we're reaching them through the NAS report. No offense.
I'll say that I think Hank does a great job of attacking a view out there that's a prominent view, and that we need a public consensus before we move on, and just kind of talking about the vagaries and how difficult that idea of consensus is. To me, what's impressed me the most has been attempts at deliberative democracy experiments in various countries, where you're basically taking citizen juries or other methods. My colleague Arkan Fung does a lot of this work. There is a big one, a global one, that's being planned, I think, for this year or next year in Athens, which should be very interesting in that respect. But all that said, you should only ask the public for their opinion if it's something you think the public's opinion matters, right? For people who think there's either a right of scientific inquiry or this is a constitutionally protected right of procreation, rights claims are such that they trump kind of this view of the public. I'm not sure I believe quite that, but I think that this is kind of a bit of a disconnect of what we ask the public for and what we don't. And it's important to know what we're asking and why.
Michelle, I would just like to briefly add that
yes,
in the immediate aftermath of JK's appearance in Hong Kong, one of the leading scientists who was in the room told me off the record that he felt that the whole JK affair in his in germline editing was an existential threat to people like him who are leading or running or launching or founding companies such as Editas, CRISPR Therapeutics, this whole wave of somatic gene editing companies trying to provide therapies and cures for sickle cell disease and a long list of other disorders. Thankfully, the two have not been conflated, at least not so far, by politicians launching committees and inquiries. And so we hope that books and efforts and films and all the public engagement that, as George says, many scientists do commendably, will help ensure that at least the people who make the rules are sufficiently well informed.
Well, I think that's a great note to end on, and we are out of time. There's obviously so much more I'd love to talk with everybody about, but I want to thank all of our panelists for joining us today and giving us such a rich set of perspectives and deepening our understanding of these issues. And especially want to thank Professor Greely for giving us this wonderful book. Again, the next time you are in quarantine or just sitting with a really good cup of coffee, it's a wonderful way to spend an afternoon. So thank you very much. Thank you to Professor Myler for bringing us together, and please look out for future announcements of events in the faculty book series. Thanks everyone for joining us.
Thanks everyone.
Everybody.